Hello students. Earlier the video was abruptly ended. So we are discussing on the substrate concentration. So whenever the substrate concentration increases or decreases, this is one of the factor which affect the enzyme activity. What we were discussing in the last part of the earlier video, that the substrates, first of all we need to increase, and whenever we increase the substrate, then first of all the velocity of the enzyme increases. But at one period of time, whenever the substrate concentrations are further increased, then the velocity of the enzymes remain constant and it already attains its maximum velocity. Okay, so first of all, here we have written by increasing the substrate concentration, the velocity of the enzyme first increases, but increases at one point, further increase in substrate do not change the velocity of the enzyme and attain its maximum velocity. Okay, so what we can call that, first of all the substrate concentration we need to increase. So we have a constant number of enzyme. First of all, whenever we are increasing the substrate, then the velocity of the enzyme increases. But as we are going on increasing the substrate concentration, then further increase of the substrate do not increase the enzyme velocity, and at one period of time velocity of the enzyme become constant. So what is that? That will be called as Vmax. Okay, so here this point we will show it in a graph. Okay, so let's just see, whenever we try to show it in a graph, this specific sentence we will put it in a graph. Okay, so let's just see what will happen. So what happens, first of all, this is the graph here. Okay, so first of all what we have done here, we will take the substrate concentration and here we will take the velocity. Okay, so first of all, in this, first of all we are increasing the substrate, then the enzyme velocity will also increase. At one period of time, whenever further increase in the substrate do not increase the velocity, that means the enzyme already attains its maximum velocity. Okay, so that will be called as the Vmax. Okay, so this will be called as maximum velocity of the enzyme for a specific concentration of the substrate. So what is happening here, at what point exactly of the substrate concentration the enzyme got its maximum velocity, we don't know. This may be this may be this may be this, isn't that? So that's why what we have done, that we have taken half of its substrate. Okay, so whenever we are taking half of the substrate, then the half of the velocity it will get it attain. Okay, so this half of the velocity will be represented as half Vmax. So at a specific concentration of substrate, the enzyme will attain half of its maximum velocity. So what we can represent it with, that specific substrate concentration will be represented by a constant. That constant was developed by two scientists, Michaelis and Mendel, and this constant will be called as Km. This is called as Km value, which is constant for specific enzyme, and this was given by two scientists, Michaelis and Mendel. So that will be called as a Km value. Okay, so I hope you understand about this graph. So this is very much important, right? Then after that, let us move to the next part, that what happens if you compare two enzymes, right? So if you compare two of the enzymes and both of them, they are having the same Vmax, one have more Km, one has less Km. Which will be a better enzyme? That means which is a faster enzyme to produce more product, that which have less Km. Okay, so if you need to see, then I can show it in a graph. See, we are going to have suppose two enzyme. So this is the enzyme 1 and this is the enzyme 2. For both these two enzymes, which is going to produce more product, right? So here we will put two graphs. Okay, here this also have this much and this is also have this much of Vmax. Okay, so this is the Vmax. For both of them, this is supposed to be same. Half of Vmax is same. One, suppose this is the Vmax. Okay, now here the Km, half of the Vmax will be this much, right? So this will be the Km and for here this will be the Km, right? So this is the Km. So if you compare, I have not using any scales, so maybe like this, but if you randomly, if you just go through roughly, then you can see Km value for this enzyme is more and Km value for here is less. Okay, so if the Km value is less, okay, then this enzyme will be a better enzyme. That means the velocity of the enzyme will be faster as compared to this first enzyme, and that's why it will go or show more velocity, so it will produce more product. So this may also can come. So this is also one reference of the earlier graph. Okay, so this is about the type of enzymes, how the factor, that is substrate concentration, affect the enzyme activity will be this much. So what we have seen, these are the three types of enzyme activity. We have seen different factors we have discussed. One was the temperature, another was the pH, and the third one is the substrate concentration. Okay, then after that we will see inhibition. Okay, so on this part we will see the types of inhibitors. Okay, so what are the inhibitors? They are structurally, by shape they are similar with the inhibitors and the substrates. Okay, they are going to have the same shape, so they can compete for the same active site for with a enzyme. Okay, so we are discussing the first one, that is competitive inhibitor. Inhibitor means which is decreasing the enzyme rate, or they are decreasing the product, or their reaction is decreasing. So this will be called as inhibitor. There are two types of inhibitors, or there are two types of inhibition. One is competitive inhibition and another one is the non-competitive inhibition. So in this competitive inhibition, what is it? See, some of the inhibitors are present. Those inhibitors have the same shape with the substrate, but obviously they are different. Okay, so they are chemically different molecules. Okay, so just see, this is one enzyme, this is the enzyme, this is one substrate. So this is the substrate here, so this is substrate, this is the enzyme, and what is it? This is the inhibitor, right? Suppose this is the inhibitor. Okay, so as we have seen here, this is the inhibitor, this is the substrate. So active site here, this is the active site, okay, of this inhibitor, and this is the active site of the substrate. So both of them, they are same in shape. So both of them, they will compete for the same active site with the enzymes. So that's why what happens, what we can get, that both the inhibitor as well as the substrate, both of them will try to make bond with the, that means they will try to get attached to the enzyme. Obviously, when inhibitor bind to the enzyme, then obviously it will be loosely attached because chemically it is different, and I have told you that enzymes are very much specific. They can attach to only one kind of substrate. So this inhibitor is going to be attached with the enzyme. They will not allow the substrate to be attached. They will just blocking the active site, but it can never convert, or it will never produce one product. Okay, so substrate can attach to the enzyme and convert it into the product, but whenever inhibitors are trying to bind with the enzyme, then it will not allow the substrate to attach. So that's why the product formation, how much product should be present or produced, that product number of product to be produced will decrease. Okay, so in presence of inhibitors, okay, what will happen, the product formation decreases. Okay, and how can we overcome the this inhibitor, competitive inhibitor? Whenever we increase the substrate, then the product formation will also increase, because we are allowing less inhibitor to bind with the substrate, or we are increasing the competition in between the substrate and inhibitor. If you increase the substrates, then more and more substrates will try to bind with the enzyme, and inhibitors will get the less chance to get attached to the enzyme. So yes, whenever we increase the substrate, then yes, the activity of the enzyme or activity of the inhibitors can be decreased. Okay, so competitive inhibition can be overcome by increasing substrate concentration. So whenever we increase the substrate concentration, then what happens, the competitive inhibition can be decreased, or we can see more product formation. So this can be also graphically represented. So let us see how this graph look like. After that, see, whenever it comes to the competitive inhibitors, how it can be graphically represented. Okay, so first of all, whenever there is no such competitive inhibitors, then we can see the normal graph, as on this graph we have already discussed, the Vmax graph. So whenever we add, or we have, or there is a presence of competitive inhibitor, then obviously the competitive inhibitor as well as the substrate will try to make, or try to compete for the same active site and try to bind with the enzyme. So that's why the Vmax, if you see this graph, if you see, then yes, they will attain the same, the Vmax, but what we have to do, that to inactivate or to decrease the action of the competitive inhibitors, we have to increase the substrate concentration. So the substrate will be required more to overcome the maximum rate of the reaction, or that product formation. So you can see, in presence of the competitive inhibitors, what happened, the Km value increases. Okay, so remember one thing, in presence of the competitive inhibitors, there is no effect on the Vmax. Vmax remains same, and only the Km value increases, right? So this is the competitive inhibitors, right? So there is one example what we have. There is succinate that will convert to fumarate. In the biology, in the botany part, you have already seen it. Succinate convert into fumarate in presence of one enzyme. That enzyme is succinate dehydrogenate. Now succinate and malonate, both of them, malonate is a competitive inhibitor, so malonate as well as the succinate, both of them, they will try to compete for the same active site to bind with the succinate dehydrogenase enzyme to convert into product. That succinate will try to convert into product, but this malonate will try to make a bonding with the succinate dehydrogenase, and after that fumarate production will be decreased. So this will be called as the competitive inhibitor. Okay, so competitive competitive inhibitions we have seen. After that, let us move to the next, that is non-competitive inhibitor. So what is the non-competitive inhibitor? Actually, see, this is the enzyme. Okay, this is the enzyme. This will be the substrate. Okay, this is enzyme, this is the substrate. Both the enzyme and the substrate, they have the same structure to fit with the enzyme. That means this is the active site with which the substrate can be attached. Now, what is it? Non-competitive inhibitor will be completely different. Suppose this is the non-competitive inhibitor. They're not going to have any type of this non-competitive inhibitors will be even not in similar in shape with the substrate have a different shape. And suppose this will be the inhibitor, in this non-competitive inhibitor will be completely different in the shape also. Both the substrate and the non-competitive inhibitor will not be competing for the same active site to bind with the enzyme. So this non-competitive inhibitor will try to they will try to add with the enzyme, but in any of the another sites, but not in the active site. They will be attached to any of the pockets or any of the crevices, but they will not bind in the active site. That means there is a no competition between the substrate and non-competitive inhibitors. Okay. But there is a very dangerous thing will happen, that in this case, when this non-competitive inhibitor will bind with the enzyme, then the active site will be changed. Suppose this will be the active site. So active site is completely different, suppose. Okay, so whenever the enzyme converts its active site, then the substrate will not be able to bind with the enzyme. So this is very dangerous case in case of animal kingdom. In presence of non-competitive inhibitors, our biochemical reactions can never occur. So what happens, the non-competitive inhibitors will be different in shape with the substrate. So substrate and non-competitive inhibitors are not going to have any competition to bind with the active site. The non-competitive inhibitor will bind to the enzymes in any of the another site other than the active site and change the active sites of the enzyme. So that's why the substrate will not be able to bind with the enzyme. So if this enzyme is not able to bind with the substrate, then what will happen? The product formation will be declined. It will be not even declined, this will be completely goes down to zero. There will be no product formation takes place, and if there is no product formation takes place, obviously that reaction can never happen. So even if you increase the substrate concentration, decrease the substrate concentration, no one is going to decrease the activity of the non-competitive inhibitor. So there is one example. We have one, suppose already you have got the ETS. In this ETS, we know that how the cytochrome oxidase help in the ETS electron transport system. Now what happens, there is a non-competitive inhibitor present called cyanide. If the cyanide is present in our body, readily ETS will be stopped and this individual dies, because it completely converts or it completely blocks all the cytochrome oxidase. So that's why ETS reactions cannot occur. Okay, so if you increase the substrate or decrease the substrate, this is not going to change any activity, or there will be no change in the product formation. Product formation will decline to zero. So this can be shown or graphically represented by like this. Okay, normally this is the reaction. In presence of non-competitive inhibitors, obviously this Vmax is going to change. So if the Vmax is changing, then what will happen? Here, this is the Vmax. The Vmax will be changed. Vmax will be decreased, but Km remains same. In competitive inhibitors, what we have seen, in presence of competitive inhibitors, Km remains same and Vmax, what happens? Okay, so this is going to be for non-competitive inhibitor. What happens in non-competitive inhibition? Km remains same and Vmax decreases, right? But for competitive inhibitors, what will happen? The Vmax remain same Okay, Vmax remains same, and what will happen? Km decreases. Okay, so this two will be required. Okay, so much about this will be not needed. There will be some sulfur drugs will be also there in some books you can get, but it is not described in NCRT, so that's why it will be not needed. The last type of inhibition we can see, that is feedback inhibition or allosteric inhibition. So this is not exactly a type of inhibition, or it's not a type of inhibitor, but here the normal reactions are going on, like the substrate in presence of in presence of enzymes convert it into a product. So what is happening? See, there is a reaction. We have glucose and ATP in presence of hexokinase. It converts into ADP and glucose 6-phosphate. So this is a normal reaction. So in this feedback inhibition, what happens? This hexokinase decides how much product will be produced. Okay, so product decides itself how much product should be produced. So if product formation occurs or product is increased, then the rate of the reaction will decrease automatically. And if the product formation is very less, then the rate of the enzyme becomes faster. Okay, so this will be called as the allosteric inhibition or feedback inhibition. So that is all about the types of inhibition we have seen. The last part we left, that will be the classification of enzymes. So let's just move to the classifications. Yeah, the classification of enzyme. See, there are thousands of enzymes are there. All of them, they are classified under 6 categories, 6 classes: class 1, class 2, class 3, class 4, class 5 and class 6. The class 1, there are some odd lily you can remember. So what is it? You cannot change the classes. One can never go to the last. Okay, first of all, we have the oxidoreductase. So we cannot move this class 1 to suppose class 6. The last one is class 6 is ligase. You cannot change the ligase to class 3 or class 4. Okay, a lot of people don't know about it, but yes, you have to remember that the classes of all the enzymes are fixed. It is given by, it is decided by international community of there is one society where they have the scientists grouped the different thousands of enzyme in 6 categories, in 6 classes. Let's just see the classes. So after that, let's just move to the last part, that is classification of the enzymes. So in this classification of enzyme, you just remember O, T, H, L, I, L. Okay, so there are 6 classes of the classification. There are 6 classes where we are grouping the thousands of enzymes. So let's just see, first one is oxidoreductase. Most of the reactions, biochemical reactions occurring in our body are oxidation and reduction. Okay, so what is happening in oxidoreductases? Some parts will be oxidized, some parts will be reduced. So some examples we have, that is dehydrogenase, oxidases, etc. Next one, we have the transferase. Some groups will be transferred from one to the next one, so that will be transaminase, which is also naturally happen in our body. Next one, hydrolases, some enzymes which will add water in it. One example is protease enzyme, which is also very much common. Next one, lyase is one type of enzyme which break the substrate. Okay, so one example we have aldolase. Next one, isomerase. There are different type of structural optical isomers they are forming, and after these reactions enzymes will be isomerase enzyme. And last one is ligase, which is going to add the different substrates, like the different RNA fragments can be joined by RNA polymerase, the DNA fragments can be joined by DNA polymerase, right? So this ligase, one example will be ligase, phosphoenol pyruvate, carboxylase. They are all the types of ligase enzymes. So this is all about the types, classifications of enzyme. We have completed the 2 parts, 2 videos. I have completed the enzyme part. The first video was abruptly ended in one part because of some error, then it was ended abruptly, and I have continued after that. So I'm requesting you people to watch both the two videos one by one at the same stress, so then only you can understand this part. Okay, the very much important part, it was just divided in the last part of the first video and second the first part of the second video. So you just please watch both the two videos together. So that is all about the types of enzymes. We have discussed a number of things about the enzymes, and in need one question from the enzyme part will be sure, 100% one question will come. So that is all about the types of different parts we have discussed about the biomolecule. This is about a whole chapter of biomolecule. I hope you understand. If you know these parts, read thoroughly the NCERT, practice the questions from NCERT fingertips. That is all about it. I hope you understand. Thank you.